This presentation is about the process of gamete formation in both the male and female reproductive systems. Recall that the cell cycle involves the process of mitosis. This is defined as division of the nucleus, and the result is two identical daughter cells. The original cell and the daughter cell are diploid, meaning that they have two of each chromosomes, or a pair. For example, in the human, there are 23 pairs of chromosomes, or a total of 46 chromosomes. This process of cell division occurs in almost all somatic cells. Mitosis is divided into phases. Interphase, which technically is not part of mitosis, but occurs between each round of mitosis and is the time when the cell is carrying out its functions. And then prophase, metaphase, anaphase, and telophase. On the other hand, meiosis only occurs in the reproductive cells. This is because the goal of meiosis is to reduce the number of chromosomes in the daughter cell, resulting in haploid cells, meaning they only have one of each chromosome. So a gamete, or reproductive cell, only has 23 chromosomes, not 23 pairs of chromosomes. There are two distinct phases of meiosis, Meiosis I and Meiosis II. Meiosis I happens in phases just like mitosis. We just put a 1 at the end to indicate that it is part of Meiosis I. This results in two haploid daughter cells. Each of these daughter cells will enter Meiosis II. In prophase I, all 46 chromosomes must be duplicated. Many images show this as an X-looking structure. Then the homologous pair lines up together during the process of synapsis. Homologous means that they are the same chromosome number, one from mom and one from dad. As they come together, they form a structure called a tetrad, 2 copies of mom's chromosomes and 2 copies of dad's chromosomes. And in this image you notice that there are 2 red and 2 blue on each of these. So the 4, 1, 2, 3, 4 is a tetrad. We could say that the red came from mom and the blue came from dad. In metaphase I, the tetrad lines up in the middle of the cell. In anaphase I, the tetrad is pulled apart. Meiosis ends with telophase I. The cells begin to separate, and each daughter cell is now haploid, with only one of each chromosome. But notice that they are still duplicated, but only one from mom or dad. Both of these daughter cells will go on to Meiosis II. In Meiosis II, there are the same phases again, just with a 2 at the end. Prophase II, metaphase II, anaphase II, and telophase II. At the end of Meiosis II, there are a total of 4 haploid cells, each with 1 copy of all 23 chromosomes. Here's a picture of what goes on in Meiosis II. Notice on the far left that there are 2 cells. These are the 2 daughter cells that went through Meiosis I. In metaphase II, the still duplicated chromosomes light up in the middle, and in anaphase II, they are pulled apart. Telophase II is the time when the new cells form, and at the end at the far right there are 4 daughter cells. Now that we have talked about meiosis in general, we will talk about any differences between the male and female and what the cells are called at each stage. Male gamete production is called spermatogenesis, and it occurs in the seminiferous tubules of the testes. Spermatogenesis does not begin until a male reaches puberty. From that time, a male will undergo spermatogenesis to some degree for the rest of his life. The process from stem cell to 4 gametes takes about 64 days. If the entire length of the seminiferous tubules went through meiosis at the same time, a male would only be fertile every 64 days. However, each section of the tubule has cells in each stage of spermatogenesis to ensure a constant supply of mature spermatozoa, or mature sperm cells. The original stem cell that begins this process is called a spermatogonium. It is located in the basal compartment of the seminiferous tubule. This spermatogonium undergoes mitosis, and one stays in the basal compartment and the other will enter the luminal compartment. This second cell is now called a primary spermatocyte. The primary spermatocyte will undergo meiosis I as it travels through the luminal compartment and will produce two secondary spermatocytes. Both secondary spermatocytes will go through meiosis II. At the end there are 4 spermatids, each haploid with 23 chromosomes. A spermatid is an immature sperm cell. It must go through the process of spermiogenesis to become physically mature. Here we see a breakdown of each phase of spermatogenesis. The spermatogonium at the top undergoes mitosis and sends one of its primary spermatocytes into meiosis I. The primary spermatocyte goes through meiosis I and produces two secondary spermatocytes. These go through meiosis II, and the end result is 4 spermatids. After spermiogenesis at the bottom, there are 4 mature spermatozoa. You notice that spermatids do not look like what we can think of as sperm. That's why they have to go through the spermiogenesis process. This image shows the basal and luminal compartments with sperm in various stages of development. So this is part of one loop of a seminiferous tubule. These are the interstitial cells. This is a nurse cell. And so we have the basal compartment, which has our spermatogonium. Then this would be a primary spermatocyte, and then we go on primary, secondary, and then we have the spermatids. And then spermatids, they're not quite completely mature yet. Female gamete production is called oogenesis. It occurs much the same way as spermatogenesis, with some slight differences. At the end of oogenesis, there is only 1 ovum or egg produced instead of 4. The cells still undergo division of the DNA, but the DNA from the other 3 cells is put into small cells called polar bodies. Oogenesis occurs in the ovary, and another difference between genders is that it begins before birth. The stem cell of oogenesis is called an oogonium. Mitosis occurs before birth, and a primary oocyte is produced. This primary oocyte will begin meiosis I, but stop during prophase I. Until puberty, nothing happens, and these primary oocytes stay in the ovary in what are called egg nests. Each primary oocyte is in what is called a primordial follicle, and at birth, each female has about 2 million of these primordial follicles. Through a process called atresia, most of these primordial follicles will degenerate until about 400,000 are left at the time puberty begins. Once puberty begins, the female pituitary gland begins to secrete follicle-stimulating hormone, or FSH, each month. As the name implies, FSH stimulates a few of the primordial follicles to develop. On the inside, the primary oocyte will complete meiosis I. The 2 cells produced are called the secondary oocyte and the first polar body. This secondary oocyte begins meiosis II, but stops at metaphase II. At ovulation each month, it is a secondary oocyte in metaphase II that is released. If the female is not sexually active or if fertilization does not occur, the secondary oocyte will never finish meiosis II. However, if fertilization occurs, the secondary oocyte finishes meiosis II, producing 2 cells, the mature ovum and the second polar body. This image shows each phase of oogenesis. At the top we have the oogonium undergoing mitosis and sends one primary oocyte into meiosis I. The primary oocyte goes through meiosis I and produces two cells, a secondary oocyte and our first polar body. The secondary oocyte will stop at metaphase or be released at ovulation, and it will stop at metaphase 2 unless fertilization occurs. And here we can see this cell being fertilized. The first polar body may or may not undergo meiosis II. Essentially, the polar bodies are just ways of getting rid of the DNA, not going into the mature ovum.